Hi everyone, welcome to the NEET course offered by Insight Education. My name is Madhuri and I will be taking part of your biology syllabus. Today we will start with the chapter Cell, which is chapter number 8 in your 11th standard syllabus. Okay, but before we move ahead, let me give you an introduction to myself. My name is Madhuri Parmar and I have studied MSc in biochemistry. Along with biochemistry, my subsidiary subjects were zoology. And as you can see from this slide, I have a keen interest in everything that is science. Okay, you can see me here looking through a microscope, handling an earthworm, or separating DNA from different samples. These are the plates where I used to work with bacteria and fluorescent bacteria even. Okay, so it is going to be an interesting journey. And once again, welcome. Um, I always start a new batch with my favorite quote from Albert Einstein. He said, everyone is a genius. Okay, even you all. I have that firm belief that all of you are a genius. Okay, so everyone is a genius, but if you judge a fish on its ability to climb a tree, it will live its whole life believing it was stupid, right? So, with this quote, I just want to say that now you have that added advantage over other students. Okay, that you have already given NEET once, so you know what to expect, you know where you are lacking. And the third thing, you have joined this course, which means you are ready to work hard, you are ready to give your 100%. Am I right? So do not worry, leave your worries to us, your teachers, and we will do our best to boost your score in the next NEET segment. Fine. So without further ado, let us start. The unit number 3 in your 11th standard syllabus is cell structure and functions. Okay, so we will be starting with the most basic portion in any biology textbook, which is cells. Fine. Now this unit comprises of three different chapters. The first chapter being cell, the second chapter is biomolecules, and the third chapter is cell cycle and cell division. We start with the first chapter of this unit, which is the cell, the unit of life. The NEET syllabus for chapter 8. By now, I'm sure you must have known that the NEET authorities or NTA, they give out a syllabus for each year, or at least for some years they have given out, where the syllabus is mentioned for specially for NEET. Okay, so whatever is mentioned in the syllabus, only that much if you cover, you will be able to ace it. Fine. I always tell my students that if you consider 100% in NEET, okay, then minimum of 80% is from your textbook itself, is from your NCRT. Okay, the proportion has increased these days, so it is up to 90%, but the minimum value I can tell you is 80%. Fine. So if you have 100 questions, 80 questions will be asked only from your NCRT textbook. Isn't that great? Second thing, do not focus on that extra 20%, okay, which is not even 20 these days, but those 20% are somewhat difficult level or out of syllabus kind of questions. But if you focus on that 20%, you might lose out on the easy 80% questions, right? So the first focus should be textbook, and this time around, your way should be problem solving approach. Okay, instead of just keep on reading the textbook, keep on solving more and more questions from a given topic. Okay, and that is how you will be prepared for NEET next time. Clear? If you want to contact me, I am available anytime. You can text me, ask sir for my number, and you can be in touch. Okay? Right. Now let us come back to the topic of the day. We will be starting with chapter 8, cell, and the NEET syllabus from this chapter includes cell theory and cell as basic unit of life, structure of prokaryotic and eukaryotic cell, plant cell and animal cell, cell envelope, cell membrane and cell wall, cell organelles, their structure and their function. It includes the endomembrane system, which is made up of these organelles, which are endoplasmic reticulum, Golgi body, lysosomes, and vacuoles. Then mitochondria, ribosomes, plastids, microbodies, and finally the cytoskeleton, cilia, flagella, centrioles, and nucleus. So these are all the interesting topics we will be studying in this duration of time. Okay, like any other topic, let us just begin with the history. Okay, how the cell was discovered, what is a cell, all these basic questions. Fine. Now, I believe that if you want to build up something, you have to start from the base, which is why we will be covering all the topics, so you do not have to worry. That is my portion. I will cover all the basic topics, then progressively make them advanced. Fine. And what your homework is, what your job is, to keep on attempting questions from the lectures that I have already taught you. As soon as I finish with the lecture, your your homework is to solve questions related to that lecture. Clear? Fine. So we begin with the history, how the cell was discovered. In 1674, there was a scientist called Antoine van Leeuwenhoek. Okay, or you can just call him Antony van Leeuwenhoek. Now, this Antony van Leeuwenhoek, you can see him sitting here in his study. He was the first one to observe nanomolecules. Okay, he called them nanomolecules. Now, what are these nanomolecules? So, these nanomolecules are actually living cells. And where did he observe these living cells from? He observed them from pond water and scrapings from his teeth. So he was a very curious fellow. Okay, any scientist has that basic level of curiosity. So he also always had that curiosity. He used to go and pick up random things and try to look these things under the microscope. So one time he went, he got the pond water. Okay, wherever the water has been stagnant, he got that pond water and he tried to look it under the microscope. Okay, look at it under the microscope. He saw a lot of things moving around in that drop of water. The next thing he did was he took some scrapings off his teeth. Okay, as we all know, we need to keep on brushing, right, every day. Otherwise, there is a lot of buildup on the teeth. Even in the commercials, you would have seen, if you have seen Colgate or Pepsodent or Close Up, you know they show on the teeth there are some spiral bacteria, right? So what he basically did was he took some scrapings of his teeth and then he saw that under the microscope and he found that something was moving around even in this microscopic size or microscopic drop. Fine. So he called them nanomolecules, meaning tiny, tiny organisms that were moving around. Fine. He used this simple handheld microscope to view the samples. This is the microscope he used. This is the handle, okay? What you see him holding here in the diagram. So that is the handle he used to hold. Then on this flap, you have a lens. Okay, lens is normally a magnifying glass, what you have already, you might have already seen and even studied in optics, physics. Okay, whenever you study physics. So there was a lens here, a sample holder. Sample holder is where you place your sample. In this case, his sample was pond water or scraping from teeth, or even he used to look at sperms, okay, from different animals. So all of those samples were kept here. Fine. Now you can see these two screws, right? So these are called knobs or screws. By twisting this screw, you can focus on this particular sample, whatever sample you have placed. Using this screw, you can pull it up or push it down, okay? Align it according to the lens. Then you have another knob, which is the focus knob. This knob or this screw will take this sample closer to the lens or farther away from the lens. Okay, so this is the basic kind of microscope he had been using. What is done is, now this microscope is held vertically, and on the opposite side there should be a light source. This light could either be from a lamp or it could be the natural sunlight. Here you can see Antony van Leeuwenhoek sitting in front of windows. So the light entering from the windows will enter the lens and will enter the sample, and from the sample you will be able to see through the lens. Okay, you see through this lens and the object will be magnified. Fine. Antony van Leeuwenhoek is also known as father of microbiology. So that is one important point you should consider remembering. Before Antony van Leeuwenhoek, there was another scientist. His name was Robert Hooke. Now Robert Hooke used microscope to observe a thin slice of cork. Okay. Now this is the exact diagram of the microscope he used. As you can see, it is different from Antony van Leeuwenhoek's. This is the cylinder. Here is the eyepiece from where you see. This is the sample holder. Here is where you place the sample. And this is the light source, which is a flame. Okay. And from the light source, the flame, there is a lens which will converge all of this light at this one specific spot. I assume or I hope you have already handled a microscope at some point in life. Okay, if not, just go on YouTube and check how the microscope is actually used. Okay, what are the different parts? Some of it we will be studying next, but the practical applications you can go and research on your own if you are interested. Right. So what is this cork? Cork is actually, if you have seen in the movies, right? If you have seen in movies, there are wine bottles and or even medicine bottles, and on that wine bottles these corks are used, which are called booch in Gujarati. Booch marelu chhe bottle ma. So that booch is actually made out of cork, and cork is nothing but it is soft wood. Okay, so he used cork. He sliced it thin and एकदम पतली पतली slice करी, and then he looked it under the microscope. He saw an arrangement of small boxes. This is the diagram from his book, Robert Hooke's book called Micrographia, and in that book he drew this. Okay, he hand drew this whatever he saw from the microscope, and you can see here he was able to see small boxes. Okay, and because these boxes looked like small rooms or they resembled small rooms, he called them cells. Even today you might have heard jail cell, right? Somebody is locked up in the cell. So small rooms are called cells, and from that he named these small boxes he saw in the cork also cells. The only difference between Robert Hooke and Antony van Leeuwenhoek is Robert Hooke saw the dead cells. Okay, in the wood from the cork, he saw dead cells, cells that were already dead, no nucleus or anything. While Antony van Leeuwenhoek saw living animal cells and even bacteria which were moving around. Okay, so he saw the live cells, while Robert Hooke, in this cork observation, he saw dead cells. Right? Robert Hooke is called father of cytology. Wherever you hear this word cyte, C-Y-T-E, it means cell. Okay, cell just in a site, the prefix or it could be either suffix. Fine. So because he developed this cell biology, which is also known as cytology, he is called father of cytology. Clear? The study of cells is called cytology, or in some parts nowadays recently it is called cell biology. Right. So microscopy, it is important to know that the advances in microscopy, okay, the advances in microscopes has led to new and new discoveries and better understanding of the cells. Fine. So this is a basic compound microscope. Compound microscope meaning there are two lenses which are used. Okay. In Antony van Leeuwenhoek's experiment, he used only one lens. So that was a simple microscope. Even you can use that if you have that magnifying glass. Okay. So that magnifying glass is a normal microscope. While this compound microscope means it has minimum of two different lenses. Okay, this is a basic microscope. Here is the eyepiece where a lens is present. Okay, here is where you look from. Then you have a rotating nozzle, okay, rotating nose piece, which has different magnification glasses. Okay, it could either be 20x, 40x, 60x. X means times, how many times. So 60x lens will be magnifying the sample 60 times. Fine. So those kind of lenses are present here because they will be present near the sample. They are called objective lens. Then you have a stage where you place the sample. Then you have a illuminator, which is the light source. Okay, Antony van Leeuwenhoek used either natural sunlight or lamp. Same way Robert Hooke used a lamp, and today we use a lamp, but an electrical lamp. Okay, or we even use a concave mirror to focus the sunlight towards our sample. So the light will be falling from beneath, and the sample will be lit up. Then you have two control knobs. This coarse focus will take this stage up and down, and the fine focus will make very minute changes till you can focus. Okay, you must have used mobile phones to click pictures, so you know what focus is, right? You have to focus it near or far. There is autofocus present in your phones, but in case of microscope you have to focus using your hands, okay, according to your eye. This is the basic structure of a microscope. Now, what is magnification? I've been using this word so many times. So, magnification in formula, if you want to consider, what is it? It is the size of image seen with the microscope. Okay, the size of image you see with a microscope divided by the size of image you see with naked eye. Fine. The same thing can even be used for a telescope. Okay, with naked eye the size of moon might be, let us say, 1 centimeter, but after using the telescope you are able to see moon as big as, let us say, 10 centimeters. So the magnification would be 10 times, 10 by 1. Clear? So magnification in case of microscope is also the same. And as I already told you, these objective lenses have different magnification powers. They tell you if it is 20x, that means it will magnify 20 times, 60x, 60 times, 100x, 100 times. Okay, so these are the different objective lenses. After studying this normal microscope, let us understand what all types of microscopes are there. Okay, it was not only one type of microscope that is used in science. So you have this normal compound microscope where you use visible light. Okay, it could either be a light from lamp or it could be natural light to see the samples with high magnification. The magnification value is very high, but the resolution is low. Resolution, again, you must have, when you look at different videos, or if you even if when you download different videos, you know they come in different varieties. Okay, for example, 144 pixels, then you have different 360, 1080. So what are these values? These are nothing but resolution, how clearly you can differentiate between 2 points. So this compound microscope has high magnification, but the resolution is not very good. There are different subtypes of this compound microscope. You could have phase contrast. In phase contrast, um, have you ever tried changing the contrast in pictures? When you change the contrast, the black and white balance changes, right? So you see either more depth or less depth depending on the contrast. Same thing happens here. This sample is mostly unstained. Okay, to look at different samples, you either have to stain it. Stain it meaning you have to add some dye or some stain to make it colored. Only if it is colored, you will be able to see it clearly. In case of phase contrast, you do not require to color the sample. Okay, just by the difference in refractive indices, you will be able to see the structure. Okay, so that is phase contrast. Always remember the meaning of contrast, that is the black and white balance. Okay, and the depth that you see in a picture or a sample. Fluorescence microscope uses fluorescence. Okay, you must have seen this fluorescence as well, or at least phosphorescence you must have seen. What are normally known as radium. Okay, they are not radium, but in normal life they are called radium. Okay, there will be different strips, or you might have used those stars and planets on the top of the ceiling, right? A ceiling upar eva stars and planets lagada mate eva stickers avta. And when there is light, you do not see anything. But when as soon as you turn off the light, those stickers, they look green in color or different colors, pink in color, yellow in color. They give off light, right? So such kind of fluorescent dyes can be used, and fluorescence can be used to um, uh, magnify and like look through the samples. Okay, so fluorescence microscopy is used wherever you have used a fluorescent dye. Then confocal microscopy. Confocal actually uses two different lenses, and it gives you what you can say, layer by layer. Okay, layer by, have you ever used the burst option in the phone? Okay, while taking a photograph, the burst option, burst burst option takes at least 30 pictures at a time. Okay, एक साथ 30 picture पाड़ी दे लंबा general when you long press. So all those pictures will have something different. એમાં કંઈક ને કંઈક different હશે. So confocal microscope શું કરે છે? So what it does is it takes pictures layer by layer. For example, if this is the sample, it will be taking a picture first at this layer, then second at that layer. Okay, layer by layer it will take pictures, and these pictures can be seen directly on your computer. So these confocal microscopy was used or and is used, in fact, to study different tissues, how cells are arranged layer by layer. This is all about compound microscope and visible microscopy. Then you have a second variety, which is electron microscopes. Electron microscopes do not use light. Till now, whatever we have studied, use light. Okay, a light source ne use करे છે, electromagnetic radiation. While the electron microscopes use electron beam. The source is electron beam. Okay, sorry, the what you can say, the illuminator is something which generates the electron beam. There are two types of electron microscopes: scanning tunneling microscope and transmission electron microscope. You might have heard of these. Now what happened was, visible light has some drawbacks, okay, some limitations, that up to a certain level you can magnify an object using normal light, but after that limit you will not be able to see anything which is smaller. I'll give you an example. You must have heard of ribosomes. So using this normal visible light microscopes, you cannot see ribosomes, okay, they are very small. So to see these smaller molecules and smaller organelles, electron microscopes are used. What they do is, here scanning tunneling microscope will use electrons, and you can see the sample's surface. Okay, only the surface of the sample will be can be seen using this STM. Okay, in 3D. So for example, the surface of your hand, you will be able to, you can see, right? There are fingers, and then near your knuckles, it goes up. Then you have different veins where the skin is up, right? So that is your surface. The same way, the surface of your cell or surface of your sample could be seen using scanning tunneling microscope. Transmission electron microscope is used to see the internal structures of a cell. Okay, what is inside a cell, what all particles are present, then the cell membrane, it is so thin, so that is also used to TEM is also used to clearly see the cell membrane. So this is all about microscopy. um, as of now, this is not a part of your NCRT syllabus, but this is just an extra. Okay, sometimes it gives you an extra edge over other students, other NEET aspirants. You need to have that extra something, okay, to clear NEET. So apart from basic, like I already told you, we will also be discussing a few advanced things which you should know. That it was just because of development in microscopy that you are now able to understand each and everything about a cell. Okay, without these microscopic advancement, you will not even be knowing what a cell is made up of, right? So it is so much important. Now, what is the size of a normal cell? So here is a scale from zero point 0.1 nanometer to 1 kilometer. The atoms are zero point 0.1 nanometer or angstrom. They are measured in angstrom. Then you have amino acid, proteins, viruses, chloroplast, and finally around 1 micrometer. Micro means 10 raised to minus 6. So around 1 micrometer to 10 micrometer or micron, you have most of the bacteria. Till so from atoms till the subcellular organelles, you have to use electron microscope. Okay, which is very clear, and you can observe things that are smaller up to 2 nanometer. Okay, 2 nanometer or even less can be observed using electron microscope. Then cells you must have seen, or if you have not, do not worry, we might encounter it someday, but we normal plant cells or animal cells from your cheek and from onion, you might have seen them under the microscope. So that is the normal light microscope you used. Okay, so from 10 micron to 100 micron, you have plant and animal cells which are all very visible. Okay, from chloroplast or subcellular organelles till the um, eggs or even plant cells, you can observe under the microscope, light microscope. You do not require electron microscope for this. Then you have ants or any other insects and all the things from 100 micron. Okay, from 100 micron or 1 millimeter to n kilometer, you can observe with your naked eye. So from 100 micrometer till the longest distance you can see with your naked eye, with your human eye. So for this you do not require a microscope. This is how the size of cell and how the electron microscope or light microscopes are used to um, see what different kinds of samples. Now let us come back to the history. In 1835, so long after Robert Hooke and Leeuwenhoek had already seen cells, and Robert Hooke had also named them cells, in 1838, there was a scientist, a German botanist. Botanist is a scientist who studies botany, right? Plants. So Matthias Schilden was working with a number of plants. He was looking at different plants all the time. And he observed that all plants are composed of different kind of cells. Okay, all plant tissues are made up of different cells. Today you already know what all kind of cells they could be, right? When you study plants, you might have already studied what all different kind of cells are seen in a plant. Okay, then there was another scientist around the same time, okay, in 1839, there was a scientist, a zoologist called Theodore Schwann. Now, how to remember his name is this Schwann actually sounds like a swan, like SWAN, swan. So swan is a bird, hunts, right? And so from that name, you can remember that he was a zoologist. If you want to remember, that is not an important part. For now, why I'm focusing on the zoology part is because he observed animal cells. Okay, so because he was a zoologist, he studied different type of animal cells and reported that they have a plasma membrane. Okay, he could see that there is some lining around the cell and he called it plasma membrane. He also saw some plant tissues and plant cells. Okay, he had a microscope, he was looking at different animal cells. He thought, might as well look at some plant cells. So when he compared, he saw that plant cells have an added cell wall, which the animal cells do not have. So that was something unique to the plants, he said. Together, Matthias Schilden and Theodore Schwann, they gave the cell theory. Okay, the cell theory means that all organisms, plants and animals, are made up of cells. Because they looked at a lot of plants and a lot of animals, and they found that they found that all of them were made up of cells. So they give this first statement that all organisms, plants or animals, are made up of cells. And cell is the basic unit of all living things. Anything which is living is made up of cell. Okay, it is a most basic part of anything that lives. Now there was a problem, like they were not able to answer a question which asked that how do these cells arise? Okay, where do they come from? Where do the cells come from? Imagine yourself in 1839 and somebody asks you, okay, if every a plant and every animal is made up of a cell, where is this cell coming from? So this was the question they were not able to answer. There was another scientist after 15 or 20 years called Rudolf Virchow. Okay, in 1855, Rudolf Virchow explained that cells divided. Okay, he was the first one who saw the cell divide. He saw one cell getting divided into two cells, and from that he said that new cells are formed from pre-existing cells. Okay, the mother cell will divide and give you two daughter cells. So that is what Rudolf Virchow said. He used the term omnis cellula e cellula. Okay, so cells arise from pre-existing cells. He gave the final shape to the cell theory, and now Schilden, Schwann, and Virchow, they gave a unified cell theory, which is made up of three statements. All living organisms are made up of cell and are product of cells. Cell is the basic unit Okay, structural and functional unit. All cells come from pre-existing cells. Okay, the cells which are already existing, from that the new cells are arising. Clear? So till now, this is the end portion of today's lecture. What did you understand? Okay, so if somebody asks you a question, what is a cell? What did you understand from today's lecture? So cells are basic structural and functional units of life. Okay, if you break a cell, if you go one level below the cell, it is no longer a living thing. Okay, for example, if you just study organelles which make up the cells, organelles cannot be called living things. Okay, so cells are the basic structural and functional units of life, like brick in a wall. Okay, brick is the structural unit of a wall. Same way, cells are structural units of life. They are the smallest living factories. They are the smallest living things, okay, and they have a lot of metabolism going on inside them, a lot of chemical reactions which keep you alive. So they are the smallest living factories. That is what you came to know from today's lecture. And third part is the size and shape of the cells may vary. Okay, you already have studied that a neuron, okay, a brain cell is very different from a cell from your liver. Okay, a hepatocyte or a liver cell is very different from your neuron, right? And it is very different from a, let us say, bacterial cell. So the size and shape of cells may vary, which means they may be different, but their basic composition remains the same. What they are made of remains the same. So what is this basic composition? If you take a pie chart like this, 70% is made up of water. Okay, so can you see here I've just taken an example of a bacterial cell. The same is true for human cell as well. About 70% is made up of water. Then you have 30% which is made up of different chemicals. What chemicals? So these chemicals includes protein, then small molecules which can be sugars, amino acids, different metal ions, different negatively charged ions or anions. Okay, all these small molecules, tiny molecules are also comprising a cell. Then you have RNA, ribonucleic acid. You have studied tRNA, rRNA, mRNA. So RNA, then you have phospholipids. Phospholipids make up your cell membrane. Then organelle membrane is also made up of phospholipid. 1% is DNA, which is found in your nucleus. And a very less proportion is of polysaccharides. Polysaccharides include starch, glycogen, cellulose, and pectin, all of these. Fine. So now you know what a cell is, what it is made up of, and what the cell theory is. However, these three statements of cell theory have some exceptions. Okay, some exceptions. What are these exceptions? The first one is organisms of kingdom Monera. Monera means bacteria. Okay, kingdom Monera. You must have studied the five kingdom classification. If you have, if you do not remember, do not worry. We will come across it some or the other time. However, kingdom Monera means bacteria, and Protista. Protista are unicellular eukaryotes. So these two are actually acellular. Okay, what does acellular mean? So acellular means, especially in case of Monera, they do not have many organelles. Okay, they have only one single cell doing all the function. Even in that, Monera does not have cellular organization. Okay, it does not have cell organelles. It is just a capsule inside which there is nucleus and there is there is nucleic acid, which is DNA, and cytoplasm. So these both are considered acellular. Because they are acellular, or because you cannot consider them as proper cells according to Matthias Schilden or Theodor Schwann, they are exceptions of the cell theory. Then RBCs, your red blood cell, sieve tube cell of phloem, these all do not have nucleus, okay, and other vital organelles, but they are still living. So these are also exceptions to the cell theory. Third is viroids. Viroids are even smaller than viruses. Viroids are basically just RNA. Okay, they are infectious RNA. So you have viroids, then virus. Virus is made up of a DNA or an RNA covered by protein. So it is a nucleoprotein. So virus, prions. Prions are again proteins, okay, but they are infectious proteins. For example, here you have a normal prion protein and then it becomes a disease causing prion protein. Nobody knows exactly how this is happening or what makes it get converted into this disease causing prion. Fine. So these are subcellular structures. Subcellular matlab smaller than cell, okay, lower than cell. So these are subcellular structures and they are not even non living, okay, they are considered non-living because they do not follow the cell theory or what a cell should be made up of. They do not have nucleus, they do not have cytoplasm, they do not have other machineries. So they are called subcellular non-living structures and they are exceptions of the cell theory. Multinucleated organisms. So there are some organism or some cells which keep on dividing, their nucleus, but do not divide the cell. Okay, so the cell remains common, but the nucleus keeps on dividing and the cell keeps growing. Example is rhizopus, which is a fungus. Then Vaucheria. Vaucheria is the largest cell you can find, okay, largest living algal cell that you can find. Just go and Google Vaucheria, you will see they look as big as grapes, okay, that huge of a cell. So Vaucheria, which is a big algae. Then human skeletal muscle cell. Right, the skeletal muscle cell you must have studied in movement and locomotion chapter. So that skeletal muscle cell is a very long fiber and it has many number of nucleus. So all of these are exceptions to the cell theory. Okay, and finally, the first ever cell to exist on this earth, Prithvi par, on this earth, the first ever cell, the first ever life did not come from a cell. So that cell is also an exception of the cell theory. It did not come from a pre-existing cell, right? So today we end the chapter here, the lecture here. Fine. If you have any doubts, please let me know. And second thing, you will be provided with different material for the same chapter. Keep on solving the questions, okay? You keep on solving the questions. If you have doubts, we can discuss. Otherwise, solving questions is the only answer, okay? That is how you can develop the problem solving approach. As we move ahead and we will complete at least half the portion, we will also start solving some advanced level questions or important questions in the lectures itself. Okay, since this is the first lecture, we end it here. Okay, see you next time.